and I just want to give you a quick overview over the technology and also a few teasers on what we've achieved so far. Just as a side note, because some of our technologies are currently being applied for patents and are in the pipeline to be published, I can't go into too much detail on some of the things, but I think the overall technology I will be able to explain to you. So when we think about drug delivery, when we think about how we treat modern diseases, we have a few standard ways that we inject drugs, that we target them in, or that we bring a drug into the human body. So obviously you can use something like an eye drop in the eye. You can use mucoadhesive sprays. Pills and capsules are probably familiar to everyone who's here. And we have injections, we have gels. We have all these ways that we can bring drugs into the system. However, they're usually not really targeted inside the human body. Most of these ways always rely on a very systemic exposure to the drug. This means that we essentially pump a lot of the drug into the human body, and then we just take a step back and hope that some arrives where we want to have it. And because of that, we usually give the body a lot higher dosage of the drug than we would ordinarily need. And this results in adverse reactions and side effects. The idea behind the ANGIE project is that we want to build a micro delivery platform which we can use to bring drugs into the human body in a very targeted manner. So you can see a little video of this here. This is an example where we can see that inside the human body, we can have these microrobots, and they can go to where we actually want to have the drug, and we can really target them at one specific area. In the ANGIE project, we concentrate, in order to manipulate these structures inside the human body and to control them, we concentrate on magnetic navigation. There are different technologies out there in the field, and we think magnetic navigation is going to be the most promising, mostly because magnetic forces have very nice scaling properties. So even if we go to small robots, we can still control them pretty well. We have, with magnetic fields in general, we have good biocompatibility properties over a wide range of conditions, which means that the human body is almost transparent to magnetic fields, at least in the range that we want to use them. We can use magnetic fields to generate heat. For example, this can be done via hypothermia, where you use high frequency oscillating magnetic fields, and magnetic fields are really controllable. In general, there are certain methodologies that we can use to locomote things inside the human body, or locomote things with magnetic fields in general. We can use gradient fields. We can use, as you see in this robot here we have a little rod which we locomote with gradient fields and then manage to manipulate cells. We can use oscillating magnetic fields, as you see with this robot called a magmite. And then we can use the rotating magnetic fields. So as you can see here, due to the inherent structure of this artificial bacterial flagella, we are able to locomote it using rotating magnetic fields. In the past, we've already shown a few approaches to using these inside living systems, or first of all, in vitro. As you can see here, we have this little microrobot delivering a drug to these cells. And we've made incredible progress over the last couple of years when we talk about building systems that are large enough to fit a human inside. Here you can see one of the first versions for this, and we also within the ANGIE project now made a lot of really cool progress in the direction of getting these really economical and into the hospital. The field of microrobotics is about 20 years old, and in this time we really solved a lot of the issues that we had. And the idea behind the ANGIE project is to now make this final push that we need in order to take the technologies that we have in our labs, that we usually use on the microscope, that don't really exit our lab space, and really push it into the hospitals. And for that, we have a few long-standing challenges that we really have to think about. and that we can only solve together. And this is also one of the cool things that we have in the ANGIE consortium, is that we have so many different backgrounds. We have people with a very good background, for example, in the field of pharmacology, in biology, in microfluidics, and only together we will be able to solve this challenge. As you can see here, this is an image of a movie which I think was released in the 60s, Fantastic Voyage, and already there people imagined that one day we can go into the human body and actually do these drug delivery and operational tasks from within. And we are definitely getting closer to this, and it's the goal of ANGIE to achieve this. If we look at the goals that we have with the ANGIE project, we want to deliver relevant therapeutic amounts. We want to be able to control the rate of drug delivery. We want to use it in realistic volumes. This is something that has been neglected a lot in the literature, but obviously if you want to do it inside the human body, you need to have a workspace that is large enough. We want to have a system that is compact enough to be integrated into operational rooms. Now there comes a lot of the economic aspects into play. For example, we need to think about how hospitals work. We need to think about if you can actually run this, or how we can make this technology very economical. We need to comply with regulations, obviously, otherwise we can't use them inside the human body, or we will not be allowed to do this. And also by doing all this, we want to minimize the physical burden that we have on healthcare professionals, for example, from exposure to X-rays. For now, and I think this is something that we also want to discuss in the working groups, but for now, the ANGIE technology really focuses on strokes. So strokes are the second leading cause of death and the third leading cause of disability. And worldwide, every 2 seconds, someone will have a stroke. It is estimated that 1 in 6 people over their lifetime will have a stroke. So we can already see that we might have a few stroke cases, unfortunately, in this meeting even. Just as a simplified explanation, what do I mean if I say stroke? Specifically, I mean the ischemic stroke. So a stroke in general is the blockage of the brain vasculature resulting in a sudden cell death. Obviously, if a brain vessel inside your brain is blocked, you can't get the blood there. The blood delivers the oxygen that the cells need to survive, and that won't be possible. In an ischemic stroke, especially, we have this blockage in the brain vasculature, and ischemic strokes are the majority of strokes that we have today. The standard way of treating strokes is to use a drug called TPA. And TPA is a protein that really opens up these brain vessels. In practice, when we treat patients with TPA, we always need to make a trade-off. So we want to give a drug, a dose, because nowadays we inject it intravenously. So it goes everywhere in the body. So we need to find a dose that is high enough to dissolve the clot, but on the other hand, not so high that the patient suffers from very harmful side effects. Or we can also say, all right, we accept some of the side effects, which are not too bad, in order to open up the blood clot. And here you can only see some of the swellings that RTPA can cause inside the human body. So from the ANGIE technology, what we really focused on over the last couple of months is to fabricate a microrobot technology, which actually looks a little bit different than this. But this is a good example photo. We also investigated the load and release kinetics of our drug. We investigated together with our partners in Crete the biocompatibility aspects of this, and we optimized our entire system. So in the end, now we already have first iteration of our drug delivery robot. We really thought long and hard about the materials that we want to use, and soon there will be also a paper about this out regarding the magnetic materials, especially that we want to use. We optimized our magnetic working conditions. We are already looking very deeply into the magnetic field generation devices that we want to install in the hospitals. We are thinking very hard about biocompatibility and biodegradability in order to make this whole system actually work inside the human body. And obviously we need the proper drug loading.